Regulation of transcription and translation. The effect of oestrogen on gene transcription. We've learnt previously how hormones such as adrenaline, which are protein-based hormones, are able to use second messengers to have their effect on the transcription and translation of proteins within the cell. In general, the principles involved in controlling the expression of a gene involve controlling transcription. So for transcription to begin, the gene is switched on by specific molecules that move from the cytoplasm into the nucleus. These molecules are called transcriptional factors. Each transcriptional factor has a site that binds to a specific base sequence of the DNA in the nucleus. When it binds, it causes this region of DNA to begin the process of transcription. Messenger RNA is produced and the information it carries is then translated into a polypeptide. When a gene is not being expressed, the site on the transcriptional factor that binds to DNA is not active. As the site on the transcriptional factor binding to DNA is inactive, it cannot cause transcription and polypeptide synthesis. Hormones, like estrogen, can switch on a gene and thus start transcription by combining with a receptor site on the transcriptional factor. This activates the DNA binding site by causing it to change shape. Oestrogen is a lipid-soluble molecule and therefore diffuses easily through the phospholipid portion of the cell's surface membranes. Once inside the cytoplasm of a cell, oestrogen binds with a site on a receptor molecule of the transcriptional factor. The shape of this site and the shape of the oestrogen molecule are complementary to one another. By binding with the site, the oestrogen changes the shape of the DNA binding site on the transcriptional factor, which can now bind to the DNA. It has become activated by the oestrogen binding to it. The transcriptional factor can now enter the nucleus through a nuclear pore and bind to a specific base sequence on the DNA. The combination of the transcriptional factor with DNA stimulates transcription of the gene that makes up the portion of DNA. And in so doing, oestrogen is able to switch on the transcription of a particular gene. In the absence of oestrogen, the transcriptional factor cannot bind to the binding site on the DNA and cannot cause transcription. In some cases, it is thought that oestrogen may cause the transcription of genes involved in uncontrolled cell division, and in so doing, cause tumour formation. And in this case, it's possible that oestrogen may be linked to the development of breast cancer.